Molecular Oncology, ICMR, New Delhi. The topic of her presentation is Tumor Tissue Proteome Analysis of Early Stage Gallbladder Carcinoma.
Surgical resection along with the radio and chemotherapy and targeted therapy are the options. Generally, this disease is diagnosed at the early stage because of various reasons such as the vague symptoms, its anatomic position, because it is present below the liver, it is many times not detected in the radio imaging, and the lack of screening methods or the molecular tests for early detection of the disease. So therefore, generally it is detected at the advanced stages, and which leads to the poor survival of the patients. So my lab is working, carrying out various research activities aiming towards identification of circulatory biomarkers for early detection of the disease. So we are doing tissue proteomics, and we are also working on autoantibody response analysis, where we can identify the tumor-associated antigens which lead to the production of antibodies which can be detected and which can be used as a circulatory marker. And we are also working on extracellular vesicles proteins from the blood plasma, and which again can be used as the circulatory markers, and also directly analyzing the plasma proteins. Today I will be sharing the work where we have done the tissue analysis of the early stage gallbladder cancers. This is showing the TNM staging of gallbladder cancer. So early stage gallbladder cancer includes stage 1 and 2, where the tumor cells are restricted to gallbladder. Whereas in case of advanced stages, the tumor cells disseminate to the nearby organs and leads to metastasis. And the survival outcome, as you can see, is much higher in stage 1, which is 80 to 100%. Whereas in the stage 2, it is 28 to 75%. And in case of advanced stages, it is much less, which is around 15% overall. So we have done the tissue proteome analysis using the stage 1 samples and stage 2 samples. So we have taken the tumor tissue from the controls. Here the controls were used, the GSD, gallstone disease cases with no dysplasia or metaplasia were used. And in the first experiment, we had used GBC stage 1 cases, and we performed iTrack based quantitative proteomic analysis. Similarly, in another independent experiment, we have used the controls and the stage 2 cases. And after performing the iTrack based quantitative proteomic analysis, we identified the differentially expressed proteins in both stage 1 and stage 2, which led to the identification of non-redundant list of differentially expressed proteins in early stage gallbladder cancer. So this led to understanding of the molecular process and pathways associated with early stage gallbladder cancer, and further we have verified some of the key regulatory proteins. This is showing the clinico-pathological parameters of the patients used for the study. So overall, we have used 12 patients, which included 7 patients from stage 1 and 5 patients from stage 2. This is showing the SDS page profile of the tissue lysate from early stage gallbladder cancer and controls. So as I mentioned, in the experiment 1, we have used stage 1 cases. So we have taken, here we had pooled the tissue from the GSD cases, and we had pooled GSD cases. We have pooled 6 cases where we have used the similar tissue weight from all the cases, and we had taken the individual sample from the gallbladder cancer cases and did the ITRAC labeling, followed by pooling of all the samples, followed by the SCX and C18 cleanup. And further, we performed nano-RP LC-MS analysis and then identified the proteins and quantitated them. Similarly, we did the experiment for the gallbladder cancer stage 2 cases. This is showing the PCA plot analysis. So here you can see this is for the stage 1. So GSD is showing in one of the quarters where like 2 of the cases were showing somewhat similar profile to GSD, whereas the other cases were showing relatively different molecular profile. Similarly, for the stage 2, again GSD is which is control, is showing in one quadrant. However, the other 2 of the GBC cases were showing in the same quadrant, whereas the other 3 were showing quite different molecular profile. Whereas the technical replicates, which are showing in different colors, red, green, and blue, are showing similar good correlation. So this is showing the differentially expressed proteins in early stage gallbladder cancer. So overall, we identified 357 proteins showing differential expression in early stage cancer, whereas here like 83 proteins were detected in both stage 1 as well as in stage 2, whereas 101 proteins showed up only in stage 1 and 173 proteins showed as differential only in stage 2 cases. Out of these 357 proteins, we did the literature survey and we found that 68 proteins were showing already they were reported to be differentially expressed in gallbladder cancer, whereas 272 proteins were reported to be differentially expressed in other cancers and 17 proteins were novel to cancer. So already the transcript data was available for gallbladder cancer. So we compared our proteomics data with the transcript data. We found 97 proteins mapped with the transcriptome data, and 73% of the proteins showed positive correlation in expression at both protein and transcript level. This is showing the gene ontology. So we found majorly the proteins were located in the cytoplasmic proteins, and we also found the membrane proteins, nuclear proteins, and extracellular space proteins. And we performed the pathway analysis using the upregulated differentially expressed proteins as well as the downregulated differentially expressed proteins. Among the upregulated proteins, we found neutrophil degranulation pathway related proteins to be overexpressed, while extracellular matrix proteins were downregulated. This is showing the volcano plot where the differentially expressed proteins were showing up. Okay. This is the hierarchical clustering analysis. Here we found that there were 2 clusters. Cluster A majorly included stage 1 cases and cluster B included stage 2 cases. And we found cytokeratin proteins to be overexpressed while they were downregulated in cluster B. So we found 29 proteins which were showing to be differentially expressed in more than 6 patients. This is the list of 26 proteins. And when we performed the protein-protein interaction analysis, we found neutrophil degranulation proteins were clustered separately, and ECM proteins were showing a separate cluster, and the cytoskeletal proteins were showing a different cluster. So they majorly we found overexpression of neutrophil degranulation associated proteins, which included neutrophil marker proteins such as myeloperoxidase, CEA, CAM8, ITGAM, and integrin B2. And these are the other proteins, neutrophil degranule proteins. And these are the downregulated proteins of extracellular matrix and the cytoskeletal proteins. So we have come up with a hypothesis that there is neutrophil infiltration in the early stage of gallbladder cancer, and it leads to neutrophil degranulation, releasing the various proteases, leading to the degradation of ECM proteins, which in turn leads to invasion, increased cell invasion in early stage gallbladder cancer. So although neutrophils are infiltrated in the other the inflammatory conditions also, there are tumor promoting neutrophils. There are two types, N1 and N2 types. Tumor, we believe that if there is a presence of more of tumor associated neutrophils, tumor promoting neutrophils, we can target those neutrophils for the therapeutic applications.
Then please close.
Yeah, thank you. Just two more slides. So we
Come
have
to the
just last
conclusion.
slide. So we have analyzed the differentially expressed proteins for their presence of signal sequence, and whether they are reported in the serum or plasma in various cancers. So we found 51 proteins which are showing both the signal sequence as well as the presence in the serum in various cancer condition, and we believe that these may be further explored as the circulatory markers for early detection of the disease. Thank you.